Susan. Good evening and welcome to the Basic Science Lights the Way series. Many of you are old-timers at this. I'm, of course, Dean Botchan, and especially excited for tonight's conversation about the powerful connections between biology and physics. I say especially because I actually have my PhD in biophysics. We have a fantastic panel of experts who bring both theoretical and experimental backgrounds to this discussion, so it should be very enlightening. Many of the most profound discoveries happen at the intersection of scientific fields, which is why UC Berkeley is organized to encourage interdisciplinary collaborations between researchers. It's all too easy to compartmentalize academic disciplines and expect specialization in one field. But there are physical parameters to what happens at the molecular and cellular levels, and a greater understanding of how these rules lead to stronger science being performed in our labs. Biophysics can be broadly divided into three main areas: molecular, cellular, and systems biophysics, the latter describing collective phenomena such as evolutionary dynamics of populations. Each subfield applies physics principles to uncover biological processes, and our experts tonight will reveal the fascinating research that they conduct and what it means for our understanding of how life actually works. Once we're underway, I hope that you will post your questions, as Susan said, for our speakers in the chat box. We will answer as many of them as we can. So please take note of that again. Our moderator this evening is Abby Dernburg, professor in the Department of Molecular and Cell Biology here at UC Berkeley, a senior scientist at the Lawrence Berkeley National Laboratory and a Howard Hughes Medical Institute investigator, and an affiliate of the California Institute of Quantitative Biosciences. Professor Dernburg investigates how genetic information is transmitted from parent to progeny through the specialized cell division of meiosis. Meiosis is the specialized cell division process that gives rise to the reproductive cells such as sperm and eggs, pollen and spores. Errors in this process underlie many human birth defects, such as Down syndrome, and also contribute to human infertility, especially in older women. So understanding this process has meaningful implications for us all. Her research lab uses the roundworm specimens to investigate these mechanisms, which has enormous experimental advantages using these worms, including rapid and powerful genetics, robust genome editing, outstanding cytology, and the opportunity to directly observe meiosis in vivo in the actual cell that's producing these germ cells. Professor Dernburg has received numerous awards for her work, including the Edward Novitsky Award from the Genetic Society of America, an Early Career Life Scientist Award from the American Society of Cell Biology, and the Presidential Early Career Award for Scientists and Engineers. Thus, she's patently interdisciplinary in her scholarship. We're delighted to have you here this evening, Abby, as our moderator. And I'll now mute myself, hard as it is, and turn the show over to you.
Thank you, Mike. I'm really excited to be here to moderate this edition of Basic Science Lights the Way. And as Mike's already told you, tonight we'll hear from three of my distinguished colleagues about work at the intersection of biology and physics. This is something I've always been interested in. In fact, when I started graduate school, I was in danger of becoming a biophysicist. I was really interested in protein structure based on my undergraduate work at Cal in Dan Caution's lab, I wanted to study protein structure. But as often happens in my first year of grad school, I was exposed to new ideas and new methods. And I got very drawn into the world of microscopy, which is also a physical technique, although it's considered sort of a lower resolution approach to studying biology than the kind of work that you'll hear about, for example, from Ahmet. Although I never really thought of myself as a cell biologist, that's ultimately what I became. But what I've also seen happen over the last 30 years or so is that the lines between cell biology, structural biology, biophysics, and other fields is becoming very blurred, and our understanding of the problems that I'm interested in have become very molecular in nature. So I've sort of come full circle. So we're going to have talks that span sort of different scales of biophysics, starting with Ahmet Yildiz. And after each talk, there'll be question and answers. So if you have questions for the individual speakers, please, as Mike said, type them in the chat and we will answer as many as we can. And then if there's time at the end, we'll also have a quick Q&A with all the speakers. So now I'm going to introduce our first speaker, Professor Ahmet Yildiz. So Ahmet is a professor in the Department of Molecular and Cell Biology and also in the Department of Physics. His research lab combines biochemical and single molecule biophysical techniques to understand how motor proteins move or like crawl along microtubules. They can move very long distances at high speeds, and they produce pretty impressive forces, which are required to carry different kinds of cargo, including, I would add, chromosomes, along these sort of train tracks that span cells. So despite decades of extensive research on microtubules and their motors, our understanding of how motors are recruited to specific cargoes and how they deliver cargoes to correct destinations within the cell remains very partial. Owing to recent progress in biochemical reconstitution and cryo-electron microscopy, we are now starting to develop really molecular understandings of microtubule-based motors, how they transport cargo in vitro, and start tackling many of these long-standing questions. Ahmet, thank you for joining us this evening, and please go ahead and take the stage.
Thank you, Abby. Hello, everyone. Thank you for being here. Today, I will tell you about how to apply some of the physicals to understand the mechanism of molecular machines that function in your cells. So molecular machines hydrolyze the energy of ATP hydrolysis, other forms of energy inside cell to perform mechanical work for fundamental cellular processes such as replication of your DNA, reading of this genetic code to make new proteins, or sometimes recycling unwanted or unfolded proteins that are no longer functional to make new ones. So these machines use the ATP energy, and ATP is made by another molecular machine that is embedded in the mitochondrial membrane. So this machine has channels that are so thin that can only transport a hydrogen ion. And as the hydrogen ions go from one side to another side of the mitochondrial membrane, the channel starts rotating, and which is coupled to this mushroom-shaped subunit that binds to ADP and phosphate to make ATP. So most of our cellular energy is being made by this machinery. The part of molecular machines that I'm particularly interested in are the ones that actually walk along linear tracks of cytoskeleton. So this is actually a kinesin motor that is walking along the mitochondria, walking along the microtubule filaments and carrying a lipid vesicle that actually carries signaling molecules for our cellular membranes. So intracellular transport is a fundamental cellular process. In this process, kinesin and dynein motors bind to the specific cargoes such as organelles, vesicles, messenger RNAs, or other signaling molecules. And then they walk along the microtubule in opposite direction. Kinesin walks in the forward direction from center to the periphery, and dynein walks in the backward direction from the periphery towards the center of the cell. And by controlling the activity of these two motors, cell can control how to transport material back and forth from one place to another. And this process is especially important for development and maintenance of asymmetric cells, such as neurons, because neurons can be 1 meter in length, and most of the material is made in the cell body, and they are needed in synaptic terminals or dendrites. And these are actually being carried over by these tiny motors that are taking nanometer size steps. And to cover 1 meter, they need to take 120 million steps to go from cell body to all the way down to the synapse. And here in this movie, you see the transport of mitochondria, the power plant of the cell, by molecular motors along the neuronal axons. As a biophysicist, I'm particularly interested in how these motors can take steps and generate forces needed to carry a cargo. To measure forces, we use a technique called optical trapping. In this technique, we polymerize the microtubule tracks and lay them on a glass surface. Then we purify these motors from the cell and attach them to glass beads. And then we shine a focused laser beam to these glass beads. And light carries a teeny amount of momentum. And when it passes through the bead, it changes its path. Because light changes its path, its momentum changes, so it actually applies force to the bead. So if you focus a laser beam to the center of the bead, what happens is that when the bead wants to move to the right, the light pulls it back to the center, so bead actually gets trapped. So in this movie, you can actually see this is a diffusing bead in the solution, and this cross shows you an invisible focused laser beam. So bead is diffusing, it gets trapped, you take it and bring it on top of a surface immobilized microtubule filament, and motor binds and starts walking on the microtubule filament. And then what happens is that bead gets away from the trap center, but eventually pulls back. The reason why this happens is that the force between the trap and the bead increases by the separation, such that motor walks as if there is a spring attached to its back against the wall. So in this experiment, what we can actually see that the initially motor takes a step, takes another step, so you can actually measure the sizes of these steps. But then it starts slowing down as the resistive force increases because it is moving the bead away from the trap center. Eventually, motor can no longer take a step forward because the resistive force exceeds how much force it generates and snaps back to the trap center. So from this experiment, we can measure the step size of a motor. We can also measure how much force it generates. So motor takes 8 nanometer step size. That's the distance between the adjacent binding subunits. And it generates about 6 piconewton of force. And piconewton is 1/300th of a weight of an apple. It's a tiny amount of force, but it's still pretty significant at the molecular level. But we also are interested in how to see how these actually motors take a step. And beads are too large to see these things. For these purposes, I developed a fluorescence particle tracking technique that we actually attach light-emitting molecules that are 1 nanometer in size to the legs of these motor proteins or their feet. And then let's say that this is a kinesin motor, we actually attach 1 blue dye and 1 red dye. And then by tracking the position of 2 dyes, we can actually see how these motors walk. For example, this is a single channel, just one of those dye molecules. We actually take each of these images and fit this emission profile to a Gaussian function, find this in the center of the emission profile, which shows where the dye is. And by doing this for 2 colors, we can actually see how these motors take a step. So like it's the blue labeled foot takes a step, red, red, and blue, and red. This actually shows that kinesin walks in a manner similar to human walking by alternating left and right stepping. When we do the same experiment for dynein motor protein, we actually do see that the dynein actually steps in a different way. When we zoom into this trajectory, we see that this one foot can take 2 steps, 2 successive steps before its partner moves. And steps do not always generate from the rear. The foot in the lead can actually take a step and extend the lead. So they are stepping by uncoordinated stepping of its 2 feet. So I don't have an animation for this, but Monty Python has an answer to this. So dynein actually steps by misregistered steps, sideways steps. It can also take steps sideways. While this actually sounds less inefficient than the coordinated stepping of kinesin, dynein actually becomes more successful to avoid obstacles on its path because of this irregular stepping behavior. And finally, I also want to tell you a little bit about more recent application where we studied why dynein moves towards the minus end of a microtubule. Based upon the frozen pictures taken by electron microscopy, we see that dynein releases from the microtubule, and then this linker domain that connects the motor domain to the cargo binding region goes from the straight to band conformation. This band conformation is believed to push the microtubule binding domain towards the minus end. So according to this theory, direction the linker swings is the direction dynein moves. If this theory is correct, I can change the direction the linker swings relative to the microtubule to see if I can control the direction based upon the change in direction. So this is a native dynein motor. It swings toward the minus end. And this is why what I want to do is to engineer a dynein motor that swings towards the plus end of a microtubule. For this, we change the angle dynein leans to the microtubule, and then we'll flip the ring 180 degrees around this basic coil so that it will swing towards the right-hand side. So flipping the ring was pretty easy because dynein connects to microtubule by this coil. So if you elongate the helix, it will rotate. But then leaning and changing the leaning angle was pretty difficult without messing up with its function. So we actually basically find the residues that are actually creating this kink and take these neighboring residues, move them one residue at a time, one position at a time to see at which position it's going to lean backwards. So these are computer simulations. This is mutant dynein. After 50 nanoseconds, this mutant starts bending in the opposite direction, suggesting that it is the one we should actually be focused on. We take these mutations, put it into a monomeric dynein motor, and take its frozen pictures by electron microscopy. This visual histogram shows that the native dynein leans towards the plus end, as we know this before, but the engineered dynein is leaning towards the minus end as intended by our design. Then we take this mutant, put it into a dimeric dynein that walks along the microtubule. Native dynein walks towards the minus end, and which direction does the engineered dynein move? So these are polarity marked microtubules. This is the minus end, this is the plus end. Let's just change the colors and look at the engineered dyneins. And engineered dyneins move towards the plus end of the microtubule. What this experiment shows us is that this theory is correct. Linker swing generates the Nace-Weiss and dynein stepping, and linker swing is directed towards the minus end by angle and length of the stalk. So when you look into the sequence of all dyneins in nature, they all have the same stalk length, and they all have those two proton residues that generate the kink. What this suggests is that all dyneins in nature are probably minus end directed motors, and this mechanism applies to all of them. For my talk, I would like to thank the people who did the work in my lab, and I would like to thank my collaborators, and thank you very much for listening.
Thank you, Ahmet. That was really amazing. It's just incredible how much progress there's been in this area. There are some questions coming in, and let's see, I'm gonna first just ask you about the technology of the optical trapping. So how do you know that the optical trap is not sort of affecting the motor's natural ability to move cargo? How do you know that you're measuring its intrinsic force generation?
Sure. I mean, in this case, we are using spherical beads, and they are allowed to rotate around their own axis. And to make sure that the optical trap is not introducing any torsional resistance, we actually generate this flexible linkers between the motor protein and the bead so that it can swivel around without creating any barriers. And we also look at how fast the beads are being moved by optical trapping, just to make sure that attachment of a bead doesn't actually change the velocity of the motor protein. Yeah.
Thanks. Okay, so a question from Benai and Alec Dar Abrams. They want to know if microtubules are linear or do they form bifurcating networks and junctions?
Yes.
And yeah, go ahead.
Individual microtubules are linear fragments, and they're pretty rigid with respect to their molecular size. But in cells, microtubules are bundled together to form a whole bunch of different networks and structures. For example, in the case of cell division, microtubules are formed into a bipolar spindle from two poles, and they actually hold the chromosomes and then separate. In the case of axons, they actually form this thick bundle, unipolar area of thick bundles, so that they're more rigid and more stable, and motors can walk along this track without their bending and breaking.
Do motors do they stay on the same microtubule, or do they sometimes jump between?
They can jump from one microtubule to another, and sometimes they actually are rerouted, especially in the axonal branch sites, but little basic synapses that emanate from the synapses. They actually walk all the way into the axon, then make a left turn or right turn, basically go where they need to go. Yeah.
Okay. Are there any human diseases associated with defects in these transport processes?
Oh, many. Well, in cargo transport is a fundamental process. So if you get rid of this motor, actually, if you only get rid of kinesin, even dynein transport, driven transport is gone. So whole machinery shuts down in a few minutes. They depend on each other. But small problems cause very serious diseases. So, for example, neurodevelopmental, neurodegenerative diseases, and such as spinal muscular atrophy, ALS, motor neuron degeneration, and other proteins that are associated with these transport system are also causes of like smooth brain disorders like lissencephaly and Alzheimer's disease.
Thanks. This might be a longer question than you have time to answer, but George Brooks asks, how do movements down microtubule migrations interact with mitochondrial dynamics?
Well, that's a great question. So mitochondria is a dynamic organelle that can actually fuse together or split apart. So motor proteins actually play or have been shown to be playing a role in this process. It's not the ones that I'm actually showing in this basically my presentation, but there are other types of kinesins that are involved in splitting of the mitochondria. So they are actually playing a role in this process. So they're involved in mitochondrial transport, mitochondrial splitting of the mitochondria into two pieces. Yeah.
I have a question related to my own work. I'm curious. Like, we know that motors like dynein can move objects that require a lot more force than a single dynein motor can generate. So how do multiple dyneins kind of collaborate to move something that takes more force?
Well, actually, first of all, in cells, dynein are paired together. So most functional complexes contain two dynein because dynein always attaches to another large complex called dynactin, and dynactin has two dynein binding sites. So first of all, it's two dyneins rather than one. And in addition to that, multiple of these complexes can come together and transport a cargo. So there are some cargoes that are driven by 10 to 12 dynein motors and 10 to 12 kinesin motors. But in the meanwhile, even a single motor can generate an amazing amount of force. That's 6 piconewton, like 6 times 1 trillionth of an weight of an apple, is a lot of force that can transport the entire cell at a micron per second.
Okay.
Yep.
Thank you. All right, I think we have to move on to our next speaker. Thank you, Ahmet. So our next speaker will be Professor Hernan Garcia, who's an associate professor in the departments of molecular and cell biology and also physics. Hernan did his undergraduate work at the University of Buenos Aires in Argentina, and then moved to the US and earned a PhD from Caltech. The Garcia Research Group combines physics, synthetic biology, and develops novel techniques to query and control developmental decisions at the single cell level using the fruit fly embryo as a model organism. And their goal is to understand how embryonic development is driven by genes that are interconnected through complex regulatory networks. Hernan, thank you for joining us this evening, and please take it away.
All right. Well, hello, everybody. Good evening. Let's see, does this work? Yeah, there you go. Okay, so thank you for being here today. It's such a privilege to be able to share our science with all of you. And so just to get started, let me just tell you a little bit about the sort of problems that we're interested in. So we're fascinated by how a single cell can lead to making a multicellular organism. And we asked this question for the most part in the context of the fruit fly. And you see below an embryo of the fruit fly with its cells going through multiple rounds of synchronized division. Now, at 3 hours into development, you would think that, that all these cells look exactly the same to you. And yet each one of them has already decided what they're going to be when they grow up. Okay. And those decisions are manifested, for example, by these massive rearrangements called gastrulation, where physical structure, 3D structure, starts appearing in the embryo. So what do I mean by cells making decisions? So imagine that a cell needs to decide whether it's going to commit to being part of the eye or part of the leg. Well, cells inside the eye will have their genes corresponding to eyes on, meaning there will be this polymerase, this machine called RNA polymerase binding to eye genes that will make RNA that ultimately will make eye proteins that endow the cells with the properties of the eye. And on the other hand, genes that are associated with legs will be off in the eye cells, okay? And the inverse will be true in leg cells. So I genes will be off and leg genes will be on. So the main game in town that a cell uses to decide what its fate is going to be has to do with turning genes on and off. And the way that this decision to turn genes on and off is implemented at the molecular level is often mediated by these specialized proteins called transcription factors. So there can be repressors that exclude the polymerase machine from the genes, from binding to the vicinity of genes, or there's also activators that can recruit RNA polymerase to the gene such that it will make more RNA and it will make more protein. Okay, and one of the greatest achievements of biology, genetics, and developmental biology has been the identification of the activators and repressors that endow each one of the cells in the fruit fly with its identity. Okay, and people also know that these genes are organized in a network that is basically a cascade where you have a first layer of activators and repressors expressed in shallow gradients. Okay, so each cell here will be exposed to a different combination of these activators and repressors, and as a result will produce another layer of genes that are expressed in broad domains. These are also activators and repressors that will lead to the expression of genes in 7 stripes, leading to the expression of genes in 14 stripes, ultimately segmenting the embryo. And think of your vertebrae as your segments, and also endowing those segments with an identity. Are you going to be a leg segment, a wing segment, an eye segment? Now, we don't only know the molecular identities of these transcription factors, we also know the regulatory connections. So this is an amazing state of the art to start asking whether from this network topology, we could predict the developmental phenotype. Could I read this wiring diagram and tell you what the animal is going to look like at each step in its development? And to make this possible, what we do in my lab is we focus on individual arrows and we ask, given the arrangement of binding sites for, let's say, an activator near a gene, could I predict the so-called input-output function, basically the functional relationship between the output level of gene expression and the input concentration of the transcription factor? And can I do this quantitatively and so well that I can predict how this input-output function is going to change as I tune the number, placement, and affinity of transcription factor binding sites on the DNA? Okay, so how do we pull this off? Well, this is where we use our physics training, and we basically it's driven by one of the biggest traditions in physics that has to do with this dialogue between theory and experiment. And what I mean by that is that we're not just happy with cartoon models like the one I'm showing you here. We turn them into mathematical models that predict, for example, the input-output function. And then we go into the lab and we measure the input, we measure the output. And the idea is that this is a dialogue. Theory is there to make experimentally testable predictions. Experiments are explicitly designed to test those predictions and to inform the next round of theoretical modeling. Okay. Now, when we started thinking about this, it's not like people hadn't been, thinking about theory or or quantitative experiments in biology. In fact, there were all sorts of theoretical models of cellular decision making in biology. Some rooted in categories such as thermodynamic models or non-equilibrium models or models based on dynamical systems. So there was no shortage of theoretical predictions. What we were missing was the technology to test those predictions, because all of those models predict input-output functions that we couldn't measure experimentally in single cells of living multicellular organisms. And why is that? Well, there's a simple explanation. Imagine that I want to measure the concentration of my activator. So one strategy would be to combine that activator to fuse it to another protein called green fluorescent protein, GFP, that shines bright. And so you can look at the brightness as a proxy for the activator concentration. Now, the problem is that when GFP is made, it's dark. And it takes on the order of 40 minutes for it to mature and become fluorescent in workhorses of developmental biology, such as flies, worms, fish, and frog. And this wouldn't be a big deal if it wasn't for the fact that the activators we want to look at only live for 10 minutes, meaning that by the time you get fluorescence, the action is completely over. Okay? So over the last few years, we've been devoting a big part of my lab to developing technologies that would allow us to measure these input-output functions. And what I want to do next is to walk you through the technologies that we've developed in order to measure the output level of gene expression. Specifically, what we do is we go into a gene that we want to look at, and we edit it such that its RNA, when transcribed, will create a little loop. And that loop is recognized by another protein that we provide that is fused to GFP. Now, the trick is that GFP is made way in advance. So it's already bright. This does not rely on the production of GFP, but in the localization of GFP, meaning that in every nucleus in my embryo, within, let's say, a little stripe, I will have a little spot of fluorescence that will tell me how many of these polymerase molecules are making a copy of the gene. Okay, so this is what it looks like when you put it all together. In red, you see the DNA, and in green, you see the RNA signal. So I want you to notice how we start with a broad domain about this wide, and then as development progresses, it becomes narrower and narrower to end becoming a little stripe. Okay, so one of the cool things is that this is the first time anybody can see gene expression in single cells of a multicellular organism. Okay, and this is just one part of the puzzle, because I just told you we can measure the output, and you can see once again the stripe as it's becoming narrower. And the reason it becomes narrow is because there are repressors that come from the side, for example, restricting the expression of the stripe. And we can now, for the first time, see both inputs and outputs in real time in single cells. And this is the type of data we get. So here you see repressor in green as a function of time. You see how the repressor concentration is growing, slowly. And the output in magenta here, you can see how it's going through these so-called bursts of transcription, turning on and off, until the repressor concentration crosses a threshold, and then it just shuts down completely. Okay, so with these sorts of tools, we are now in a unique position to engage in this dialogue between theory and experiment that I mentioned at the beginning of the talk. And we've been making all sorts of interesting discoveries. For example, we discovered that the widespread thermodynamic models that assume equilibrium cannot account for the processes that lead to DNA becoming accessible. DNA is packed in the nucleus, and for it to become accessible, energy needs to be consumed in the context of these non-equilibrium models. We've also thought about this transcriptional bursting I showed you before, and showed that it's all over the place, and you can see cells flicker as the genes turn on and off. And finally, we've also understood how cells make stable decisions. Basically, how do they keep a memory when an activator tells it become an I cell? How do they rely on positive feedbacks to fix the decision to commit the cell to its fate? So with that, I hope that this gave you a little bit of an overview of what we do in the lab. And so this wouldn't be possible with the great students and postdocs that come here to do science at Berkeley, and without the help of these sources of funding that give us the freedom to do fun science, and with all of you being willing to engage in this. So thank you so much for being here. I'll be happy to answer any questions, but if we don't get to the end, I'll also be happy to engage over email. Thank you very much.
Thanks so much, Hernan. We have a few minutes for questions. So I guess to a first approximation, is it true that the transcription is really the determinant of cell fate, or there are other forces at work that direct cells down particular pathways?
There are many forces at work, because there's the expression whether you turn a gene on and off, but sometimes proteins can be modified. There's all sorts of regulation downstream from gene expression, but for the most part, at least in the early embryonic development of the fruit fly that we look at, the main game seems to be one of the turning genes on and off, but it's not the only one, and that's true for most of biology, I would say. There's always an exception.
What makes the fly embryo such a great system for studying this developmental decision making?
Well, from the standpoint of somebody coming from physics to try to understand how cells make decisions, it's amazing, because as you saw, in 3 hours, the cells know who they're going to be. And so you can just grab an embryo, put it under the microscope. It's really easy to image it. And on top of that, we have complete control over its genome, thanks in part now to CRISPR. We can open my computer, and like as if we were working with a Word document, we can edit its genome at will.
Okay, so there's a question from the audience. Like, what keeps these stripes so straight? Like, how do they know where they are and make a perfectly straight column of cells?
That's a great question, and one of the mysteries in biology, because basically there is an input, and the input is slowly changing levels, and one cell that sees a higher input needs to decide I'm going to go up, and the cell just next to it needs to decide I'm going to go down. So they're the answer is not entirely clear, but one of the mechanisms that people think that you can get this sort of sensitivity, ultra sensitivity to gene expression, has to do with not only one activator activating a gene, but multiple activators needing to be together on the gene at the same time. And once that decision is established, these positive feedbacks can help you fix the decision onto one fit.
Thanks. So one question is, you talk about this dialogue between experimental work and theory. Are there cases, I think you mentioned one case where the experimental work sort of revealed something missing from the theory, that you need energy to access the chromatin. Are there other ways in which the experimental work has sort of caused a revision of the theory, or vice versa?
Yeah. I mean, basically every time, many of these models come from thinking about how bacteria make decisions, and they were super successful there. And every time we've tried to contrast them with gene expression in development, we break the model. So one of the very interesting things is that again and again, we seem to be finding that you need to think about how energy is being burned or used to drive cellular processes, and that can give you all sorts of interesting things. For example, that can drive the sharp decision making that we were discussing earlier on. So yeah, I think one of the biggest interesting things about developmental biology or gene expression in eukaryotes is that it's driving both research in biology and physics, because even from the physics side, we don't really have a good way to deal with these non-equilibrium models.
Right. Yeah, I gather from our own work, it was sort of editorializing, but most of the theory of how molecules interact is, based on 1950s physics. That's all based on sort of equilibrium processes, and cells are never really at equilibrium. They're always burning energy. So
absolutely, and this might be one of the big gifts that biology has for physics in terms of pushing our theoretical understanding of non-equilibrium processes, because it's all over the place in biology.
Yep. Okay, I think we're going to hear a little bit more on that theme from our next speaker. So we're going to turn to our final talk from Professor Oskar Hallatschek. So Dr. Hallatschek is a professor in McAdams Chair in the Department of, again, Physics and Integrative Biology. His work is at the intersection of physics, ecology, and evolution. His work has provided a wealth of insights into evolutionary dynamics using both mathematical and theoretical models, as well as experimentation. His work uses methods originating from physics, and its goal is to explain how, among other things, how populations evolve in space and time. Oskar, thank you for joining us this evening, and please share your slides and take it away.
All right, thanks, Evie, for the introduction. Thanks everyone for being here. I'm also honored to have the opportunity to talk about our work. In our work, I would like now to zoom out even more to systems that consist of many molecules, not only many molecules, but also many cells and even different species, so whole ecosystems. These are really, really complex systems that are difficult and even impossible to describe at the molecular detail. However, there are always, or very often, collective phenomena of surprising simplicity, so-called emergent phenomena, that you just wouldn't see by just focusing on the molecular details. And that's kind of the focus of our interest. And emergent phenomena are not new to physics. They are classic examples, for example, pressure and viscosity. If you put a gas of interacting particles into a bottle, then you can't describe all these molecules moving around, but there's a very sharply defined pressure on the macroscopic scale, pushing against the walls of the container. Likewise, viscosity, we all know that, the resistance to flow is stronger in honey than in water. But if you look at the molecules themselves, it's very hard to see the viscosity. It's like you can't see the forest for the trees. And most exciting, I would say, in this realm of phase transitions, where the whole state of matter changes as we tune a control parameter like temperature. And it's statistical physics that has developed a really fantastic tool set to uncover a universal and quantitative rules that govern these emergent phenomena. However, all these systems that I've shown you here, they are in a way dead. They are not living. They don't constantly burn energy, as we just heard. So living systems are out of equilibrium, which makes them very, very special. And so the task here is to find what are the emergent phenomena, or how can you describe the emergent phenomena in living systems quantitatively? Are there also generic or even universal behaviors that we can describe? Now that applies also to molecular questions or questions of development, as Hernan described them. I'm zooming out and I'm interested in the emergent phenomena, ecology, evolution, and information happening on very long time and length scales. And I'd like to illustrate this space a little bit by recent work that fascinated us that's in the realm of island biogeography. So let me just give you a brief primer on that. So that really started by two eminent ecologists, E.O. Wilson and MacArthur, asking a very simple question. If you go to an island, how many species do you expect to see there? That's a simple question about a very, very complex system, an island. But they said, you know, if the island is undisturbed, maybe we can understand the number of species or biodiversity as an emergent phenomenon being generated by the competition of just two forces, one being immigration, imagine insects, spiders, and even small mammals being brought to the shores of these islands by, let's say, driftwood. And the other effect would be extinction. You know, these populations on the islands are finite, and so at a certain rate, they will go extinct. And they could show that if these two forces balance, you expect sharp equilibrium numbers of species. And so that biodiversity is kind of an emergent phenomenon that should be predictable. And that excited and energized conservation biology because people realized one should be able to apply this also to how we should create or design our natural preserves. For instance, stochastic extinction should be smaller in larger islands. And so there was the famous dictate from Jared Diamond that a single large reserve should be better in preserving diversity than several small reserves. However, it's really hard to test these kinds of theory. There were tests, but they were often relatively qualitative and often in disagreement. And there was also a lot of criticism about the theory, and research dwindled. But now the twist is, if there's really a theory of island biogeography, we should be able to also test this on a very different scale. Let's say on the microbes, on the microbial scale, where we can ask whether microbes are able to colonize their very tiny islands in a predictable way. And that's what my lab has been doing in the last 2 years, spearheaded by Yuya Karita, who was a biophysics graduate student in my group. He created, using microfluidics, a very small channel system where you can design the shape of islands that are presented to bacteria. So what you see here is a simple microfluidic channel. Here you see the scale bar, 50 microns. And as bacteria and mostly water and nutrients are flowing from the left to the right, the bacteria are seeing these invaginations here, which are our little islands. And then we are simply observing whether they're able to colonize these islands. And this whole apparatus looks like a pan flute. That's why we call it the microfluidic pan flute. And we study different species in it. It doesn't matter right now which one this is. And what we see is emergent phase transitions. So here's the first one. When we come from the small islands, we see they are mostly empty, and relatively suddenly, these cavities are being colonized. So there's a colonization transition. And what's nice is we can understand that as indeed a competition between two effects. One is birth of bacteria inside these cavities that increases this number of cells. And the other one is stochastic outflow, which is an effective death event in the system. And we can turn this into a rather precise quantitative theory that only depends on one parameter, which is kind of a critical length scale here. And from that, we can predict, for example, that density profile coming from the floor of these cavities towards the open end. And here you see the comparison with the black line with the experiments, and agrees quite well. An even greater surprise came when we looked to even larger length scales. Then these cavities became denser and denser, and quite suddenly a new phase appeared, which is here in the middle, where there's a dark phase. And if you look with a different kind of microscope, a confocal microscope, you see that there the bacteria are actually touching one another. So there's a discontinuous phase transition. We call it the jamming transition. And what's going on here is that essentially there are traffic jams emerging. And these are really collective phenomena. We know this from the highways. If lots of little errors are being made, we generate huge traffic jams. And here, huge traffic jams flipping the state of the population from gas-like to jammed. This kind of theory makes other predictions about, for instance, shock waves running through the system that are getting gradually slowing down as we get closer to the phase transition. This is also called critical slowing down. But now, so far, we just looked at single species colonizing these islands, but can we bring in additional species from the outside? It turns out that's surprisingly hard. We have to actually give the invader, which is labeled green here, an advantage to actually see any invasion. So I show you now a movie where we use an antibiotic to suppress the wild type resident, reduce the growth of the resident. You've seen that the density went down in most of these cavities, yet we don't yet see any invasion in this first day of the experiment. In the second day, we see some successful invasions in some of these cavities. Now here, clearly a successful invasion. And after 5 days, we see substantial invasion in some pan flutes, very little in others, but we never see any invasion in this new state of this living matter, which is jammed. And we can understand that by theory, because these invaders have essentially no chance of invading this population, of infiltrating this population against in this very jammed, effectively glassy state. So let me just wrap up. So coming back to this statement that a single large reserve should be better than several small reserves, in our system, that's certainly not universally true, because it depends. If the cavity, if the island becomes too large, then the population is very dense and only one species is able to survive there. It's protected essentially to an invasion from the outside. Now, this kind of interest in microfluidic island biogeography is not just of academic interest, because we actually have bacteria. We are full of bacteria on us. They live on us and in us. And they often live in tiny islands. For example, on our skins, we have sweat pores, and when the bacteria enter there, they reach into these sweat glands. And they're typically filled with bacteria of very low genetic diversity. In our colon, we have crypts that are also colonized specifically by certain types of bacteria. And so what this work suggests is that the physical structure of this environment is one way in which, for example, the host could enrich for certain types of bacteria that are important to the host. So there's potential for adaptation there. And also it illuminates how colonization resistance works, where commensal bacteria can protect us against pathogens. But I think the bigger picture here, or the bigger conclusion for me, is that there are really new types of phase transitions in these very much out of equilibrium system where proliferation is important. And we've recently written an essay about that. That, let me thank my group that has contributed to all this work for funding, and you for listening. Thanks.
Thanks very much, Oskar. So I'm curious, like you present this kind of macro scale analogy of the island biogeography, and then you kind of dive into a much smaller scale, looking at bacteria colonizing little wells in a microfluidic chamber. How do you know what aspects translate across those scales, and where the model sort of falls short of what you can say about island geography?
Well, before we do the experiment, we wouldn't really know what a transfer. That was partially the motivation behind this experiment, right? So there's this body of theory that has been developed in the 70s, and even the first book was called The Theory of Island Biogeography. And it wasn't clear that this should only apply to like the large scale of islands, right? And so we just try to see, does it apply to the micro scale? And there, however, we could do experiments, right? The other motivation of looking at the micro scale was we really care also about the microbiome and how we can describe the dynamics of the microbiome, and whether there are any generic rules that we can identify by which these microbiomes are stable and how they're evolving, which is kind of the bigger picture in our world. We want to understand the interaction between ecology and evolution.
Thanks. So kind of a related question, one of our guests asks whether, so you talked about the depth of the cavities in the pan flute
Yes.
being important. What about I mean, you didn't really talk about the other dimension, the kind of depth in this direction, and does that matter? Is that something you looked at?
Yeah, so we have varied the width of these cavities, and within a certain limit, nothing changes. But when the width becomes larger than the depth, then really the behavior changed drastically. Then it sort of doesn't, it's not anymore an island, so to speak. It's very difficult for these wide indentations to hold any bacteria. The interesting, the third dimension is a very interesting dimension, which we haven't yet looked at experimentally, but theoretically so far, we see additional phase transitions there where beyond a certain height of these devices, we see that the kind of the flow pattern changes. There are kind of vortices appearing inside the cavity in our simulations, suggesting that new types of ecological states might be appearing there. And then also you can think about varying the shapes of these cavities, which is all on the list of what we want to do.
So you didn't talk really about biofilms, but presumably some bacteria or other organisms, you know, they collaborate to develop sort of a protective environment. Is that completely not a factor in these experiments? Are you basically looking at just diffusion of the bacteria?
Yeah, so to some extent, if it's within limits, then it is part of these experiments very well, right? So biofilms, they like to stick together. So there's an attractive interaction between the different bacteria. And that's something that this kind of theory can capture. And it changes many of these length scales. For instance, this critical length scale that I introduced, upon which colonization is possible, that will be much more easily possible for biofilms which strongly adhere together and maybe to the walls of the chamber, right? So it changes certainly the parameters of our model. The question is whether it qualitatively changes the model, and that we don't know yet. Okay, there's a question here about hysteresis. Is that something, I mean, is there any hysteresis in the system or? Yes, absolutely. That's a great question. So in this jamming transition that I had introduced. Maybe you should define hysteresis actually. Oh, hysteresis means that, so I introduced this transition where when I increase the size of the cavity, at some point, the population switches from a gas-like state into this jammed state. And now you could imagine I could go back also by reducing the size of the cavity. If I could do that somehow, then the question is, do I switch at the very same size or at a different size? And if it's a different size, then we would have hysteresis. And this theory actually predicts there should be hysteresis. And this is something we could actually test by just changing the flow pattern, because the stronger the flow through the supply channel is, the deeper the flow lines enter into the cavity and the smaller the effective size of the cavity. So we could exactly go through this hysteresis loop and demonstrate that. Interesting. Okay. We just have a few minutes left, so I'm going to ask all the speakers to
come back, and if there are any questions that are sort of collectively for all the speakers or for individual speakers, feel free to type them into the chat. I don't know if anyone wants to comment. Well, actually, there's a question about the second law of infodynamics. Does anyone want to comment on that? I just Googled it, but this is something I'm not familiar with. Is this something that impinges on any of your work?
I'm sorry to say, I haven't heard that yet. I don't.
Okay.
All right.
Okay. Yeah.
Does anyone of the others know?
No.
Okay.
Sounds like a second law.
Second law of thermodynamics that is
No, infodynamics.
that is applicable to many, many other things in life,
Yeah.
not only physics. Yeah.
So there's a question about the flow rate. I think this is for Oskar. So I think how does the flow rate influence the colonization of the flutes?
So in our experiments, the flow rate essentially controls the effective size of these islands, because as we increase the flow rate, the flow lines enter these cavities, as I said before, and that makes kind of the size of the habitable region smaller. For us, that's a nice tuning parameter, that's the main effect of this flow rate.
Okay. We talked a little bit about how living systems differ from sort of equilibrium processes. Does anyone want to add anything about how the physics of or emergent phenomena differ in living systems from non-living systems?
And
It's kind of an open question.
well, I mean, the whole field of active matter, Oskar is better equipped probably to answer this question, but I would say one thing that is very interesting is that typically in physics, when we think about matter, like there's energy coming in from like in a macroscopic sense and being inserted into the microscopic world, like, think of potential energy, like, I don't know, like a water falling down a waterfall. And the interesting thing about the world of biology is that energy is coming from the molecular constituents. You saw the molecular machines that Ahmet was showing us, right? Those are burning energy, and the fact that these individual constituents are spending energy that's what leads to these weird collective phenomena that you seem to have different properties as you go up in scale. So that to me is that's why biology is so unique, and such a unique opportunity to understand non-equilibrium physics better, because it's like unlike anything else that physics has dealt with in the past.
Okay.
Yeah, maybe to add another perspective, like in equilibrium systems, you have kind of a symmetry, which is called detailed balance. And that makes it that systems in equilibrium look the same forward and backward, right? They're kind of really boring compared to life, which is very much irreversible. And so as you look at these out of equilibrium systems, you free yourself from this very strong constraint of detailed balance, and new phenomena appear that you're just starting to essentially collect. There's one field that Hernan mentioned, active matter, that has looked for a long time at system of kind of mobile agent, often mobile agents, where local ambient chemical energy is converted into movement. And people are interested in kind of the collective phenomena there, like collective motion, where suddenly you observe motion that looks like flocks of starlings moving around quite magically. And another, what I try to allude to is when you then bring in even birth and death, that also frees you from another constraint, which is the conservation of numbers of particles, because that can then change as well, and additional phenomena appear, including evolution. So I think it's a moving frontier here at this end of going away from equilibrium and freeing ourselves from very strong constraints, that we started out with, and seeing whether we can still find simple, quantitative, and generic rules to describe these systems.
Okay, so we there are a few questions in the chat, and I'll encourage all of the speakers to take a look and answer if you can. Since we're coming up on the hour, I'm going to just thank you all for your excellent presentations, and I'm going to hand the floor over to Stephen Kahn, who is the Dean of Mathematical and Physical Sciences.
So thanks, Abby. One thing I really love about this series is that I always learn something new every time I watch, even when I know more about the field or I've worked with the guest speakers. Thanks to all our panelists for describing their work in such an approachable and interesting manner. And thanks to all of you in the audience. I hope you found tonight's event informative. We'll have another Basic Science Lights the Way episode on Wednesday, November 8th, as we discuss science in the AI era, a topic that's very much in the news and is a very active field of research here at Berkeley. Please reach out if you would like to learn more about anything we covered. We'll be sharing additional resources with you by email about today's topic and speakers. And a video of this session, if you missed it or want to look at it, part of it again, should be available on our website in approximately 1 week. Our website is Basic Science Lights the Way website. You can visit that site, and you'll find there videos of all of the various sessions on a wide range of topics that we've had in the past. So you can watch any episodes you missed or rewatch ones that you liked. So until next time, stay curious, fiat lux, and go Bears. Thanks very much for coming.